Right, so this week we're going to be talking about one of my favorite topics, which is urticaria. You might know this as hives, and this is commonly sort of associated with food allergies. It's one of the early warning signs of, an allergic reaction. But really, in the context of today, we're going to be talking about it outside of food allergies and mostly as its own distinct pathology, because a lot of people don't have food allergies, but they have forms of chronic or acute urticaria. So here's a sort of overview of what we'll be doing. Like rhinitis, it has a lot of sort of classifications that we need to go through. And really, the sort of interesting thing about today's lecture is going to be talking about its pathogenesis, because it's sort of unclear at the moment, and there's some theories, especially linked to autoimmune conditions, that we'll be talking about. But it'll be interesting to sort of consider the pros and cons of that, of the available theories. So just starting off, what is urticaria? Well, this is what it looks like. These are pruritic erythematous papules or plaques. So that means itchy and then sort of red and raised welts. This is a pretty superficial swelling, so this is occurring at the sort of dermis or the epidermis. And as I said, this is a symptom of allergic reactions, but it is also its own pathology. So let's classify urticaria. Well, it can be acute or chronic. An example of acute urticaria would be during an allergic reaction. But really, the interesting sort of fields of urticaria are all going to be, in my opinion, chronic. So let's talk about how we actually classify chronic, because this one's a little more involved. So chronic urticaria is going to be taking place over an extended period of time. And if it happens with no known cause, and this does occur where, people just randomly break out in hives, this would be called chronic spontaneous urticaria or chronic idiopathic urticaria. And you'll see those used completely interchangeably. However, if the urticaria is sort of responsive or comes out of a specific trigger, we're going to call this chronic inducible urticaria. And just to avoid any confusion with idiopathic, we abbreviate it as CINDU. So this is when, again, there's a certain stimulus that directly causes the hives. And you can see in this chart here that there's a lot of these stimuli that have been documented. So things like the sun, temperature, pressure, all sorts of things. So if the sort of inducible trigger is physical in nature, then we're going to call it a physical urticaria. And I'll have a very helpful sort of flowchart at the end, but a physical urticaria is a subset of chronic inducible urticaria, which is a subset of chronic urticaria. And here are some examples. So dermographism or dermatographism is a very common one, and actually the most common one. And a lot of people, are aware they might have this. They're aware of friends that have it, but basically this is where you can, as the name suggests, you can draw on your skin. So if you were to just carve a line on your skin, it would look exactly like that top image there. We also have cold urticaria, which is the second most common. This is hives in response to temperature. We have cholinergic urticaria, which is a response to exercising. There's sort of an autoimmune basis there. Suspected the sort of sweat antigen in the skin your body is reacting to. You can see that in the bottom image. It's quite diffuse. Then we have solar urticaria, and this one's interesting because the theory is that mast cells are being activated based on certain light wavelengths, which is just really crazy to think about. So a little note on epidemiology. So around a fifth of people are going to be affected by urticaria during some point in their life, but only around 1% of people are actually going to have chronic urticaria throughout their life. So it's most common in adults, especially in men. And you can see in this map here that there are certain areas that tend to be more affected, and I'm not really sure if there's a coherent reason behind that. As far as the sort of skin goes, this is obviously the site of where all of this inflammation is happening. So I want to talk about two specific cells, or I should say point out two specific cells, which is the mast cells and obviously the Th1 and Th2 lymphocytes. So unlike a lot of atopic conditions, we see that Th1 cells are actually a little more involved here. So it's not just the type 2 cytokines, it's actually also the Th1 inflammation as well. But all of the urticarial lesions are going to show pretty strong evidence of mast cell degranulation, and unsurprisingly, histamine is going to be the central mediator here. Again, I say unsurprising because, well, histamine is the driving mediator of food allergies, and this is one of the sort of main symptoms of food allergies. So let's talk a little bit about immune signaling and how it is sort of going wrong in urticaria. So there's not a ton of data on this, but I want to bring up the P21 RAS pathway. So this is a pathway in lymphocytes, and you can see a sort of broad sketch over here. And P21 RAS is a GTPase, so it's an enzyme there that's going to be responsive to growth factors. And when it sort of goes wrong in urticarial patients, it's going to have an effect on our immune tolerance. So we're going to have an upregulation or an increase in the amount of self-reactive T cells and an increase in autoantibodies that are produced. These are going to be driving mast cell activation and the autoimmune sort of dimensions of chronic urticaria. We also see things like increased levels of IL-6, TNF-alpha, and really all that is suggesting is that inflammatory mediators are going to be a pretty reliable biomarker for urticaria, which is also not so surprising. So let's talk a little bit about mast cells and basophils before we go into pathogenesis. But mast cell degranulation, as I said, is going to be really the prominent factor occurring in all of these lesions. But actually, whether or not mast cell count is elevated at these lesions is a little controversial. So we know that mast cells are activating, and we typically mast cells in allergies, for example, might be activating through IgE-dependent mechanisms. But in these chronic urticaria patients, it's sort of suspected that these mast cells are being activated in a non-IgE basis. So there is a strong histamine correlation, but no tryptase correlation. So we're going to have to put the pieces together a bit here to understand why that's relevant. So recall that histamine is released from mast cells and basophils, but tryptase is only released from mast cells. So if we see no tryptase correlation, that would suggest that the IgE-dependent activation of these mast cells is not necessarily going to be our main suspect, and this is going to lead directly into our pathogenesis theories. So let's talk about those. There is no conclusive pathogenesis that has been established for urticaria. So what we really have is a collection of theories that are offered to explain the pathogenesis, or where this disease is coming from. So the one I want to talk about, and it's the sort of most prominent theory, is called the autoimmune theory. Now this theory basically proposes that some of these urticaria patients have an autoimmune disease that are driven by IgG autoantibodies that are targeting your own IgE receptors, or IgE molecules. And you can see in this image over here that they don't really know all of the in-between steps, but the hypothesis is that these IgG autoantibodies are interacting with our receptors and leading to those sort of characteristic symptoms. So here's a little timeline and some evidence. So initially we observed that chronic spontaneous urticaria patients had twice the rates of thyroid disease and had higher thyroid autoantibodies than the normal population. And this sort of became, offered alongside this observation that a lot of CSU patients had diseases, or there was a strong association with diseases such as lupus, rheumatoid arthritis, type 1 diabetes, which are all autoimmune conditions. So, sort of the first step here is that just noticing that urticaria, unlike all of these other atopic conditions, was really related to autoimmune conditions. In a later study, we saw that during this autologous serum skin test, or ASST, where we would inject patients with their own serum intradermally, we'd see that some of these patients would develop a wheal and flare reaction. So when someone develops a wheal and flare reaction to their own serum, it's really suggestive of sort of an autoimmune basis, because obviously the body is reacting to something within the body, right? So there's some reports as well that the sort of serum of the chronic urticaria patients has these IgG autoantibodies. Um, and we saw that when administering these autoantibodies to sort of receptors on mast cells, for example, it was able to cause histamine release in a dose-dependent manner, which again suggests that the dose of the autoantibodies is actually what's causing it. So all of this, is relatively convincing that there is some basis here, but again, the mechanism is not quite known. So let's just sort of wrap this up by talking about the pros and cons of this theory. So as far as some pros go, we see that autoimmune treatments such as intravenous immunoglobulin or plasmapheresis are actually therapeutic in urticaria patients as well sometimes. We see this really strong association with the thyroid and other autoimmune diseases, and we see a really clear identification of autoantibodies, in urticaria patients. So that seems pretty convincing, but let's talk about some of the cons. So the first one is that we have no clue how the IgG autoantibodies would actually lead to these skin symptoms. We really don't know why they would lead to symptoms in the first place, and even leading to skin symptoms in particular and not systemic symptoms is even more confusing. Also, this sort of skin serum test is going to lack a little bit of accuracy, so we don't really know, for example, if that is evidence of a true autoimmune reaction, because it might be detecting various things such as vasoactive factors in your blood or your serum as well. And the final thing is just more of a caveat, which is that just because you notice or detect these autoantibodies is not necessarily proof that they are the etiological agent. So this is more of a correlation rather than a causation thing. So it's interesting to think about this theory, and it's an example of how researchers might go around sort of forming these theories as to weigh these pros and cons. And here's this sort of flow chart that I talked about earlier, now that we're going to move into diagnosis. So this is how you would sort of document, or at least sort of diagnose which of the types of urticaria it actually is. So in the physical examination, you're going to look for the typical hives. So those are pretty easy to recognize. They're round, oval, raised, erythematous, serpiginous, all of these fun words. But if you see linear lesions, well, that's going to really suggest dermatographism above all else. So that's one of the ways you can immediately make that diagnosis. The second thing we want to talk about is history, and this is sort of the top of the flow chart. You're going to talk about how long it's been going on, so what sort of interferences it's had with daily life, and whether or not there's any specific triggers that might identify what specific type of urticaria it is. So the laboratory assessment is something really interesting to think about. So chronic urticaria does not have any confirmatory biomarkers that you can obtain via a lab assessment. So unlike things like mastocytosis, which really look for example, serum tryptase, or rhinitis, which really looks for IgE to a certain environmental allergen, chronic urticaria does not have any of those, but a lot of times people are ordering labs anyway. And the sort of logic behind that is to rule out other diseases. So there's this meta-analysis they did where they looked at all of these studies. They tried to see if there's any association between the amount of tests that someone ordered, a doctor ordered, and the amount of diagnoses that they were able to reach. And the answer was not really. So around less than 2% of these cases, they were able to identify other causes of the urticaria through a lab assessment. And you might see things like cutaneous vasculitis, for example. But really the point of the slide is that, it is an urge to be careful about being cavalier with the amount of labs that are taken, especially in populations such as children, where that might be a sort of a traumatic event. So something I want to talk about before the final area is just differential diagnoses. And if you don't know what that is, it's basically a sort of process you go through to eliminate or consider other pathologies that might be behind the symptom you're witnessing. So viral infections, bacterial infections, parasitic infections can all cause acute urticaria. So this is one of the causes of urticaria that we see especially in hepatitis. And one study saw that around 2/3 of children that went to the ED with urticaria had a viral infection. And, obviously children sort of always have viral infections, but it is interesting to think about the correlation there as well. Obviously, allergies is something we already talked to. NSAIDs, which are non-steroidal anti-inflammatory drugs, are another big one, especially aspirin. That's more of a sort of drug hypersensitivity. And then I mentioned this briefly in the last slide, but cutaneous vasculitis or urticarial vasculitis is very similar. And you would see similar urticarial symptoms, but they tend to last much longer and they're much more painful. And then also lupus is, again, going back to that autoimmune basis, is one of the areas we see urticaria as one of the symptoms. And just sort of wrapping up with treatment, beyond avoiding triggers, really the first thing that we do is give antihistamines, but around half of the patients are not going to respond very well to this. So what do you do in that case? Well, you keep hitting them with the same hammer. So you just bump up the antihistamine dose up to 4 times the daily dose. You can combine this with older antihistamines. You can add things like leukotriene inhibitors, things like allergen immunotherapy, or biologics such as omalizumab, corticosteroids. But really these is a very standard regimen, and the thing I want to point out is that these patients might be on extremely high doses of antihistamines. But with that is a sort of overview of urticaria and sort of where we think it comes from, how we handle it, how we classify it, and I hope you learned something.